Dear students, today we are going to discuss about biotechnology and its principles and process. If you take this biotechnology process, before going to discuss about this biotechnology process, let's take the term biotechnology. If you take the term biotechnology was given by Carl Ericke. So Carl Ericke was a person who was given the term or who coined the term biotechnology process. And on the honor of his work right now, we are calling him as a father of biotechnology, and the term biotechnology was given by himself. Biotechnology generally it indicates uh, mixed, or you can tell directly it's a science which utilizes properties and uses of microorganisms, animal cells and plant cells at industrial level for the generating useful products and essential to our life. And biotechnology is a multidisciplinary subject. Why we are mentioning multidisciplinary subject? Because it include all kind of subject in it. In it number of subjects can be included. So that's why we are calling it as a multidisciplinary subject. Number of institutes or recognitions were given by, or definitions were given by different types of organizations. Let us take those organizations. If you take best example, NCRT word, European Federation Biotechnology. According to European Federation biotechnology, according to European Federation Biotechnology, the definition of biotechnology now we are going to discuss. Biotechnology is a integrated use of biochemistry, microbiology, biology, biophysics, chemical engineering, and engineering science to industrial process, production of products and service. Biotechnology is an integrated use of biophysics, biochemistry, and biology and the chemical engineering, engineering science to the industrial process for the production of products and service. So this is a definition of biotechnology from European Federation of Biotechnology. And you can take the biotechnology definition according to International Union of Pure Applied Chemistry. So International Union of Pure Applied Chemistry. International Union of Pure Applied Chemistry. International Union of Pure Applied Chemistry. So according to this, they were also given definition of biotechnology. Biotechnology is the application of same biophysics, microbiology, biochemistry, and engineering sciences in order to achieve technological applications of cells and microbes. So in order to achieve technological applications of cells and microbes, that is from IUPC. And you can take United States of Nation Federation Biotechnology. So according to United States of Nation Science Federation, according to United States of Nation Federation Biotechnology. So according to this United Nation Science Federation, the biotechnology definition is biotechnology is the controlled use of biological agents such as microbes and cells and their products. So controlled use of microorganisms, which indicates United States of Nation Science Federation Biotechnology definition. And if you take this biotechnology were developed, we have been using since our ancestor time onwards. So depending upon that, biotechnology process are of two types. One is conventional biotechnology, traditional biotechnology. Second one is modern biotechnology. Traditional biotechnology, you know very well about the different types of processes, like a fermentation process, one of the best example, or conventional biotechnology methods. And here you can take different types of food products, even drying of food material, fish is one of the best example, or else you can take idli, dosha, and naan, bread, pizza. So different types of even alcoholic compounds also formation, that is since we have been using, or else you can take best example curd like cheese and ghee. So materials, and preservation of meat in honey, also one of the best example of this tradition biotechnology method. And modern biotechnology method, modern biotechnology method, the manipulation of genome was introduced. So due to this manipulation of genome, we can able to get a number of rDNA compounds or transgenic plants or transgenic products or recombinant DNA products can be obtained. So manipulation, direct manipulation of genome of cell can be takes place with the help of different types of enzymes and different types of techniques. So such phenomena we can call it as modern biotechnology. Modern biotechnology you can take in the form of two methods, that is gene biotechnology and non-gene biotechnology. Gene biotechnology, which included genetic engineering process completely or DNA technology process, and non-gene biotechnology, here you can take plant tissue culture, somatic embryos, and anther culture, that is non-gene biotechnology. So modern biotechnology are of two types: gene biotechnology, genetic engineering process or DNA technology. Non-gene biotechnology, you can take plant tissue culture methods, and you can take like a biotechnology, which is a interdisciplinary science because it included number of branches of biology. And you can take the products like a fermentation, biomass, enzymes, biofuel, and microbes, inoculants, and recombinant DNA technology, and plant animal cell cultures and process of engineering. So these all were included in biotechnology. And if you take biotechnology, it's a completely interdisciplinary science, and it was since we have been using and ancestor period onwards, and it was got more importance in 21st century regarding different types of branches. Number of branches of botany biology were included here. You can take best example genetics and molecular biology and microbiology, immunology and biophysics, biochemistry, bioinformatics, and nano biotechnology. These are completely advanced one. And tissue engineering, this is also one another advanced branches of biotechnologies were included here. And you can take this complete development of biotechnology process. And let us continue with traditional biotechnology, one of the example. So if you take traditional biotechnology, one of the example, best example, we are taking fermentation process. Fermentation, it's a biological conversion of carbohydrates or different kinds of components into the alcohol. We can call it as fermentation process. It's a Latin word. Fervere is a word which means to boil, that is indicates fermentation process. Here in this biological fermentation process was first identified by Louis Pasteur. He called it as a zymology or zymosis process or zymase enzyme, because the name of enzyme is zymase. That's why we were taken zymology process. And if you take the person that is Louis Pasteur, he was the first to demonstrate about this fermentation process. You can take different types of persons like a Cruikshank, another person Cruikshank, I'm writing here Cruikshank, CS, that indicates Cruikshank. Cruikshank, he was a person, he was also worked about the fermentation process. The term fermentation was given by himself. And Kosti Chow. So Kosti Chow, he was a person, KC, nothing but Kosti Chow. He was a person who was given this anaerobic respiration. Process of fermentation takes place with the help of anaerobic respiration. And another person, you can take best example, Buchner. So Buchner was a person who was given different types of fermentation process. So types of fermentations, we know very well about different types of fermentation. Four different types of fermentation. First one is alcoholic fermentation process, like ethyl alcohol production takes place with the help of carboxylase enzyme and dehydrogenase enzyme, alcoholic dehydrogenase enzymes. And you can take acetic acid fermentation, alcoholic fermentation and acetic acid fermentation. Here we need to know about acetic acid fermentation. Acetic acid fermentation is a acetic acid fermentation. Here acetic acid fermentation is, it is a double fermentation process. Acetic acid fermentation is a double fermentation process. And you can take another kind of fermentation, that is lactic acid fermentation process. In lactic acid fermentation process, there is no production of carbon dioxide. And you can take another kind of fermentation, butyric acid fermentation. Butyric acid fermentation with the help of Clostridium butyricum. Butyric acid formation takes place from pyruvic acid. And lactobacillus, lactic acid formation takes place. Here lactic acid bacteria or lactobacillus, it won't produces any kind of carbon dioxide. Generally, in anaerobic respiration, carbon dioxide releases, but in lactic acid fermentation, there is no releasing of carbon dioxide. And acetic acid fermentation is a double fermentation process with the help of Estobacter esti. Alcoholic fermentation. After formation of ethyl alcohol, this ethyl alcohol again undergoes fermentation process. It gives rise to the acetic acid. That's why we are calling it as a double fermentation process. This is about the entire fermentation process. Okay. Even you can take idli also, one of the best example. Leuconocentric mesenteroids. It's a name of bacteria which is involved in fermentation process. Okay. And let us take another kind of process. If you check this fermentation process, fermentation takes place with the help of vessel. So those vessels we can call it as fermenters. Generally, fermenters are very small, and in these fermenters only anaerobic conditions can be created and only microbial culture can be used. Anaerobic conditions and microbes can be used in fermentation process. Whereas if you check bioreactor, these are the another types of vessels. Here these vessels are very small. These are just 100 liters capacity, but fermenters are stainless steel 1000 liters of capacity. Here only anaerobic conditions, but in bioreactor you can take aerobic conditions as well as anaerobic conditions. Let us take here short view of fermenter as well as bioreactor, fermenter and bioreactor. If you take fermenter one side and another side is bioreactor. So I'm taking F for the fermenter and B for the bioreactors, fermenter and bioreactor. Okay. So if you take fermenter, fermenter is meant for fermentation process with the help of anaerobic conditions. So anaerobic conditions maintained. Here aerobic conditions can be possibilities as well as anaerobic conditions. Both are possibilities are there. Aerobic conditions along with anaerobic conditions. Both conditions are possible. And here only fermentation process takes place. Only fermentation process. But here number of biochemical process takes place. Biochemical processes occur. Number of biochemical reactions takes place here. And if you check fermentation process, it's a limited. The vessel is limited one, and here it is large one. It's just you can take small one. Fermenter is completely smaller when compared to the bioreactor. Bioreactors are completely larger one. Okay, here you can use only microbes as a inoculants, microbes, yeast or some other microbes as a inoculant. Here you can use plant cells as well as animal cells as a inoculants for in order to produce number of products like a pharmaceutical products, plant cells as well as animal cells. So used as an inoculant for the production of pharmaceutical products and drugs and different types of recombinant products also formation takes place. I'm repeating again, fermentation is only inside of the fermentation there is only anaerobic condition, and here aerobic conditions and anaerobic conditions can be possible. Here fermentation process takes place, here biochemical process takes place. Here it is a very small 1000 liters, here 100 liters, here it is 1000 liters, very large stainless steel vessels, and microbes are used as a culture. Here culture inoculants. Here plant cells as well as animal cells are used as a culture inoculants. If you check this bioreactor, there are two types of process takes place. Generally bioreactors contains a number of devices like pH controller and agitator and temperature controller and aeration. So different types of devices which are connected to it, and sterilization chamber also which is available here. It's a stainless steel vessels and container. There are two types of process takes place. After inoculation, we'll close the entire container and we'll do the process that is pre-process. Before getting product, what we are doing that comes under the upstream process. All the processes before starting the fermenter, such as sterilization, fermenter, preparation of sterilization for the culture medium and growth of suitable inoculum, that is upstream process. Just one word before going to switch on the fermenter, what are the process which will be takes place by for the production of number of recombinant, that is called as upstream process. If you check downstream process, after completion of fermentation process, we need to separate and segregate and distillation, filtration and addition of different types of reactant to it, and preservators and solvent extraction. So post fermentation process, that post fermentation process we can call it as downstream process. Here you can see the fermenter very clearly. Here different types of devices, motor and pressure gauge and pH controller, temperature probe and sparge for the rotation, and the impeller blades also present, and cooling jacket whenever the temperature will be rises. Here steam also can be applied. Okay, nutrient inoculants, from here only we will inoculate number of nutrients for the growth and development of microorganisms. So this is about the fermenter, most important. Let us take functions of fermenters and bioreactors. Okay, functions of fermenters and bioreactor with quick view. Okay, now we are taking another one. So applications of fermenters in industry, microbial biomass production takes place, and metabolites also production takes place. Biochemical products we can call it as metabolites, which is produced by plants, animals, and the microbe cells, produced from plants, animals, microbes. There are two types, primary metabolites as well as secondary metabolites. Metabolites are produced, maintain the life process, microbes such as primary metabolites, ethanol, citric acid, and lactic acid, best example. Secondary metabolites, you can take best example, metabolites which are not required for the vital processes, and it can be stored. You can take best example amphotericin B, or different types of antibiotics also best example, which is also formation takes place by the fermentation process. And with the help of bioreactors also, microbial enzymes, number of enzymes also can be produced. You can take protease, amylase, isomerase, lipases. And biological conversions are biotransformation, modification of substrate. Conversion of ethanol to acetic acid also can be occurs, and isopropanol to the estol, and sorbitol to the sarbose. These all can be takes place within the fermenter. And another one most important question, that is single cell protein, SCP. So this SCP also formation takes place. Single cell proteins, number of single cell proteins can be developed with the help of this fermentation process. This is about the a short view, which is helpful to the very quick revision, and in order to get 5 marks in Tamil Nadu examinations. And it is also essential to learn about NEET exam. Every time they are asking about the questions, fermenter or bioreactor. Hope you understand. Thank you so much.